So this is the second part, and we're going to discuss today second part B of mathematical modeling computational methods to define boundary between regions of the brain or region in the tissue. We will see in the third part what could be the principle of navigating in the brain. So making boundaries, we are going to show this as a conflict of two morphogenetic region for a given territories made of cells. So this is a work we did many years ago with Prochain, and I'm taking this slide from him. So if you have a cell which is expressing a morphogene A, and it will pattern activate, as we saw in the previous slide, the tissue. So on the opposite direction, you have here another morphogene, let me change color, which is called B, such as A produces own production, and so it amplifies itself, but at the same time it represses B. And so if again B activates itself and repress A, as long as you have B, the cell becomes labeled and defines one region. And so now what happened in this region where you may have the two together, which one repress the other. And then the idea is that this allows to create a number of cells that are expressing both of them in a very steep manner. So you are interested in what could define here the number of cells at the interface. Number of cells at the interface, and this number of cells defines the boundary. So this will be the boundary between two regions. So this is the idea that was promoted by Meinhardt, the late Meinhardt, in the 80s, and produced a certain number of models that was quite interesting in this morphogenetic gradient and border formation. So we can use the model I discussed in the first part, where you have the production of A, the production of these modified AK star molecules. So you have cell 1 and cell 2. You have when A crosses, it becomes A star. This A star controls this production of the new molecule A that will then pass the border of the cells and move to the next cells and get modified. However, if you have B inside, then you are going to repress the expression of A star. And similarly, if you have BK, which is produced with a symmetric manner as A, but B star is going to be repressed if you have A star in the cells. So the idea is that you have a competition for the binding site, Na plus A, which binds to this and leads to this Na star complex, and the same for B. And so you have here this expression, and you can see that when you have B, when you have B with A, you diminishes the production efficiency. Another possibility is you can have a direct inhibition, so that when you have this complex, it is, for example, destroyed. And the factor beta measures this interaction between A and B. This is what we saw here. What is the result of this if you start in one part of the tissue to express, let's say, A, in another part of the tissue you express B? You see that according to the parameter, they decay, but when you arrive to this region where they interact, suddenly the interaction is very sharp, as exemplified here, where we magnify this region. You have a very sharp decay, so that suddenly A, let me change color, A make an abrupt change in the expression. And this is quite interesting because this allows to define a very sharp region. So this is labeled as a number of cells you can see that you can have few cells that will be responsible for this boundary. And so this is another example here where we vary the different parameters. And again, you see that this is what happened when there would be no interaction. This is a dotted line. But when you have this interaction, you see that how you have this very strong depletion, and thus again this creates this sharp boundary. So you can also increase this this boundary by assuming that you can have dimer formations. That is, you need not one but two A star bound to the DNA in order for the production of A. And also this leads to some nonlinearity here in this rate, Michaelis-Menten rate function. This is for A, and this is for, in the case of bidirectional, this is the case of unidirectional. And then you are left with this equation that you can simulate. And again, so you can see with this what type of effect it leads. So it can lead to this very shallow and long gradient. It can also lead to this more decreasing, I would say, in the entire field gradient. And this is what we did for different values of parameters that are mentioned here. So I think we have I think the convention here is on the left. This is always unidirectional, and here this is bidirectional formation. So boundary formation with this dimer. So again, we were able to have even more sharper gradient, as you can see here. This is for the unidirectional case. And for the bidirectional, this was just drastic, that you don't even have the split into one. And the reason for this is that this bidirectional has to do with elliptic equation in partial differential equation, where what happened at the left and the right, it propagates instantaneously. This is the effect of elliptic. That's why you have this so sharp decay, and not much of the two steps where you have a certain decay, and then at the boundary where you have the interactions. Okay, so how can you make a shift in the boundary? So first of all, this is something that can be engineered. So this is the example of the organization of the neocortex with, for example, let's focus here on the visual cortex. So if this transcription factor is removed, you can see how this different somatosensory cortex, visual cortex, how they are going to, instead of being well organized this way, the visual cortex is suddenly going to become very tiny, and the S1 is going to take more of the space. Well, if you express this transcription factor, this is the opposite. The visual cortex suddenly occupy a much bigger space. So in all of this example, the border has changed. The region occupied, and this border has evolved. And the question is, how can we model this? So another example, this is the position of the boundary between the midbrain and the hindbrain. So this is in the normal case, you have a boundary here at a specific location. But if you change the expression of one of these OTX variant, OTX1 here, you see that there is a shift in one direction. That is, more of the cortex would be dedicated to the hindbrain. However, if you change the expressions and use this OTX2 heterozygote EN1, then you see that it goes in the opposite direction. So again, suggesting that we have this opposite way of labeling, which seeks to generate cells that have a certain identity, while GBX2 give a different identity, and the interaction create the boundary. So how can this be modeled? Yeah, and this is also a proof of these changes. So you see how here the visual cortex, if you go from C to D, you see this is direct evidences that the visual cortex region has a change, and there are a shift, you know, as you can see here from the somatosensory cortex, and more restricted A1. And what is interesting here is that if you look directly into this graph, so this we have already seen before, this is a direct example, direct picture of how you see those changes here. So this is quite clear, but what is the consequence for the behavior? So the locomotion activity has changed in the animal where you had this OTX2 knockout. And so this is shown in here, where you have a difference in the behavior of the locomotion activity. So yes, changing the patterning affect behavior. This is well known over the last 2 or 3 decades. All right, and so what we decided is to study how we can implement the shift of the boundary due to the differential initial condition. So we start, instead of having instead of starting A and B here at the same level, you start one at some level, and for the other one you have a different level. So in the case of unidirectionality, since I showed you, you had explicit expression for xk, you can then define here the shift in the boundary delta L at the position you ask, when do I get the same here, the same quantity. And so this leads then to an expression for xb as a function of xa, and you can use this to define the shift. And for example, from when alpha equals 1, when I told you that we have this 1 over, it was constant plus k, you can define the shift here as this 2dl equals this 1 over x minus 1 over xb. And otherwise, you have to use this exponential distribution. So we found out that with a change here of a factor 2, you can have a shift in about 10% for a total of 100 cells. This creates a shift of 25 cells. So this is quite interesting, because now you can see how the shift depends on the different parameter, especially of the graded expression or the parameter alpha, which account for a certain number of combination of parameters, as we discussed above. So another possibility is to see a change in the position. It's the natural fluctuation, random fluctuation on the position of the expression. So of each cells can express a certain number of A, but this A can be plus or minus, let's say, a certain quantity, and this depends on K. So if we look at each position here, you can have, instead of having this very nice deterministic decay, you can have a stochastic expression, and as a result, this will shift the boundary from where it was when there was no stochasticity to a different position of the boundary. And so what we calculated, and I'll show you this in a few slides, the cumulative effect of a flux. So you you suppose that your flux is the one that carries the fluctuations. That is, it's equal to the deterministic flux plus some, and you have to give here a model of the noise that you want to add. It leads to a shift or 2 to 3 cells, which is very different from the deterministic shift that we showed previously, which could be used to define the shift due to, for example, a missing transcription factor or an alteration of transcription factor expression. So estimation of the shift due to this flux noise. So we go back to the same equation with this is the flux. We assume here that Ka is less than 1, so we can neglect here this part. And the model is to say that the flux is phi plus a random variable where you can assume, for example, that they are delta correlated with the position K and Q. And so you can get at, using the steady state expression, you get this induction formula. And so then when you want to calculate AK, you have this multiplication by 1 plus epsilon sigma K. And if you want to see the overall effect, you have to sum and see the difference between the deterministic versus the random expression of AK. And you get this sum, and this Fg squared gives you a total effect. So it's not very hard. You sum this expression, and this tells us how the noise depends on the parameter epsilon, which is the amplitude of the noise. I think sigma is also this noise delta correlation coming from here, and alpha is give you the information about the deterministic decay of the field. So you can go to the continuum limit, and then from this you can have an expression of the flux with perturbation with sigma of x. So sigma of x, we took an Holstein-Uhlenbeck process with this exponential decay and this steady state probability, which is given by this exponential. And then you can use the bidirectional solution of the equation, which is given by this dx squared plus this flux term plus this decay term, and you end up having this deterministic solution. And then to study what happened when you have the flux, you can use this to make a perturbation. So you expand your solution into a with respect to A0, and so you have here the effect of the noise. So you can calculate the same quantities that I showed you previously, and you can expand this quantity with respect to epsilon. You can use this, so L is the length of the field, of the ensemble of the cells where they are located. The parameter lambda is coming from the Holstein-Uhlenbeck process, and basically you get this type of expansion, which again says that how the noise affect all the morphogenetic field. So estimation of the variance of the boundary fluctuations. So where is located the boundary? It is located in principle where you have the expression of one morphogene, the expression of the other one, and when there is no shift, you have the expression where U equals W. So here we do a little expansion near the middle point, and we write U as the sum of U0 plus epsilon U1. This is for the first morphogene. This is for the second one. And this is the expression of the position of the boundary with the shift. And so what you do a Taylor expansion of this along the point m divided by 2, and this is what you get. So you get the first term, the second, the first term for the 2, and then the second term in U1, and then this allows you to write an expression for the shift with respect to the expression of the graded expression of the one and the second morphogenes. And if you use the direct solution, you can have an expression for B. So again, this is done in the article. I'm not going to go step by step to each terms here. But if you have M cells and if zeta is this, has this expression, then what we found was an expression for the shift. One term is due to the noise, to the characteristics of the noise. There's Standulenbeck, epsilon, and zeta. This log has to do with decay rate, and this is the effect of the number of cells on this expression for the shift. And so the last thing I would like to discuss is now there are two cases, alpha equals 1, alpha equals less than 1. And if you start with 10 cells and we do cell division, so each color corresponds to one stage. And then when we do cell division, you change color and you see that the expression of the morphogene in a cell becomes more and more accurate. And so this is what we did. We had one cells, it is divided into two, and then we use this to simulate what happened in each cell separately and end up with a much refined graded expression here where each cell contributes to the morphogenetic field. So general conclusion, morphogenetic gradients can be generated. We show how boundary can be generated and what was the stability of this boundary. So remains a general question, is that sufficient to understand how gradients are generated in 2 and 3 dimensions? How gradients, how morphogenetic gradients, the interpretation of this gradient, how they are read at molecular levels with these DNA chromatin transcription factor interactions remains under debate, and how genetic information is transformed into a geometric organization and complex shape. This is still ongoing research projects. Cortical plasticity, how region reorganize during plasticity. This is also quite interesting because obviously the activity, brain-dependent activity, can modulate neurons and probably affect again this maybe either the gradient or the expression, the local position of neurons by pruning. And so this is still something that has not been much been modeled. Okay, and I thank you for your attention.